So I'm just going to go over a few slides to start to give a little intro of what I'll cover, but I'll mostly be demoing to you all how to do a genome-wide association study in the BioData Catalyst ecosystem. And I think we've heard a lot about all the different platforms that make up the BioData Catalyst system. And I'll try to kind of explain the part of the ecosystem that I'm working in and how that is very similar to eventually, I think, what the AnVIL will look like. Just to introduce myself, I'm Beth, and I'm a senior bioinformatics systems analyst at UC Santa Cruz Genomics Institute. And I work on a team that is working to support our partners in this ecosystem like Dockstore and Terra and Gen3, and sit in as kind of early first users to try out our ecosystem and generate content for end users, so tutorials on how to use the ecosystem, but also try to work with researchers to bring in products that they're creating and share them with the rest of the community through posting workflows on Dockstore, but then taking that to the next step of launching it from Dockstore and trying to use that workflow in a computational environment like Terra. So, oops, how do you advance on this? Okay, there we go. So just, I think you've probably seen this figure before, or you might see it again, but this is kind of an overview of the BioData Catalyst ecosystem. It has a lot of parts, and I just wanted to kind of narrow in on where we'll be working today. So in the demo, I'm gonna show you how we'll find some data from Gen3, and we're gonna export it to a Terra workspace. And I'll also show you how Dockstore workflows are used in that Terra workspace. And these three components are also represented in the AnVIL ecosystem as well. So just a quick background, if you're not familiar with a genome-wide association study, this is a hypothesis-free genome-wide scan to identify candidate genetic markers that might be associated with traits. Specifically, in our case, we're interested in diseases as the traits we're looking at. We can leverage a lot of these very rich resources that we're bringing in to our ecosystems, like what Gen3 is hosting. And these have lots of whole genome sequences paired with information about individual phenotypes. And what researchers can do is they can try to find cases or individuals that have a specific trait of interest, and we have their genomic sequence, and compare that to controls or individuals that don't display that phenotype, and try to look for places in the genome where where a specific variant or type of variant might be in a higher frequency in the cases than the controls, and this might be an area that a researcher wants to investigate further. So I'm just going to preface with, I think, a lot of times when we're trying to show these demos, they can be a lot all at once. And I'm happy for people to kind of chime in and have me slow down or talk more about a specific part of this. I think what I'm trying to do in this slide is talk a little bit about how we'll be moving through lots of interoperable platforms. So we'll be kind of changing the website. And we're also using a use case that's quite complicated, but I think really leverages a lot of what these platforms are able to do. So I'm just going to kind of quickly go through the outline of what I'm going to do today, and then we'll kind of go into actually trying it out on these websites. So I'll first open with a Terra featured workspace. It's not published quite yet, but featured workspaces in Terra are places where researchers can publish a tutorial basically that has a markdown file with a lot of information about what they'll be doing, and you can place data in that or teach a researcher how to import data into that workspace. It has Jupyter notebooks and it has workflows all associated in one place. Then we're going to move to the Gen3 part of the ecosystem, and I'll just introduce you a little bit to their graph structured data model for BioData Catalyst. I think you learned a little bit about this yesterday when Gen3 presented. We're going to search the BioData Catalyst Gen3. Sorry, if somebody could silence their mic. I'm hearing a little bit of noise. Thanks. So we'll search Gen 3 for a tutorial data set that's in the BioData Catalyst ecosystem. This tutorial data set is synthetic data that was generated from the 1,000 Genomes project, so it's completely open access. And I'm going to do a tricky thing where you can't really see most of what's in this Gen 3 BioData Catalyst stack because there's a lot of controlled access data there. I'm going to hand off that tutorial data set from Gen 3 to the Terra workspace that I'm working in, and then we'll look at what that data model looks like actually in Terra and learn how an end user would interact with those data tables, and a little bit more of what my team is developing in collaboration with GWAS researcher experts, Elisa Manning's lab at the Broad Institute. And we'll use Jupyter Notebooks to kind of interact with the Gen 3 data and reformat it into a data model that I think researchers are a little more accustomed to using. Then we'll throw that into a notebook where we can prepare our data for association testing. Jupyter Notebooks are very nice interactive features. And the data preparation step is not necessarily like a turnkey thing. It will be a place where a researcher wants to understand the data more and make specific decisions about their hypothesis before proceeding. And then we'll generate some files that we can put into workflows that actually perform these association tests. And these workflows are currently hosted on DocStore. And what I also just want to say is that this new ecosystem brings differences to how the researcher interacts with the data. So it's like a new data model. And I think the goal of this tutorial that my team is making is trying to get a researcher used to using this ecosystem and this data model, as well as learning how to do an association test. But I think going through this can help a researcher be able to use a lot of the other featured content that is on Terra that they might feel a little bit more hesitant about how it actually works when you're bringing in the BioData Catalyst or AnVIL system to these this content that's already available and has been used in kind of different ways. So with that, I'm going to move over to our featured workspace in Terra. I've kind of just used the AnVIL version to show you what this looks like. So in the featured workspace, we have a really nice dashboard that has information about what we're doing. And this is the place where I'm kind of trying to help the researcher bring all of these different components together into one area that they can follow without having to bounce around to different documentation. It does link out to documentation that we're creating about the ecosystem and how it works with all these different parts. So the first part of this is that we're going to navigate this multi-platform environment or ecosystem, and there's a few steps the researcher has to do to get their Terra account linked up to Gen 3 and so on. I'm going to skip that for now and mostly focus on moving straight into Gen 3. So we're going to tell the researcher how they can actually bring data into this tutorial. So if I clicked a link over here, it would take me to the BioData Catalyst Gen 3. And the first thing that I just want to kind of go over again, we learned about it a little bit yesterday, is that Gen 3 has this graph structured model. And I think this is a new concept to a lot of end users and maybe to many of us. And it's going to be a large learning curve on the user. And so what we're trying to do is create a lot of documentation and tutorials around interacting with data that's structured in this way. And I think this again is for BioData Catalyst, but this is also something that I think the AnVIL is planning. And so I just wanted to everyone to kind of get a chance to see what that really looks like and how a user would work with that in Terra. So the graph kind of starts at the top, and the researcher kind of moves down. The top of the graph is more administrative pieces of information about specifically the program or project. So for example, in BioData Catalyst, this might be the top med project, and then a specific study in the top med project and all the participants or individuals in that study. And if you kind of click on that, you can see the path there. So these are important links as we move down. Today, I'm gonna, if you guys can't see this, just let me know, but I can narrow in. Today, I'm just gonna focus on interacting with this section of the graph, which covers the clinical data or the phenotypic data. One thing I just want to point out before we export data to Terra is that in an association test, we have different phenotypes that we need to explore. We have the phenotype that we want to test in the association, but we also have potential phenotypes that co-vary with the phenotype we're interested in, and we need to account for that. So the researcher will really want to explore different data that's available. So if they wanted to explore, say, how something in this demographic data table compares to, say, something in a laboratory result, how do they, there's no connections here kind of laterally, so they'll have to understand that they have to go through the subject node of this graph to be able to compare these two. So there's a piece of information in the subject node that we can use as the key to join all of the different clinical nodes together. And as we're not going to go over it today, but as you move further down this graph, we'll kind of need to understand how to link the individual's phenotypic data to their genomic data. And that again through these connections here as you follow the lines down. But for the sake of time, I'm not going to go into that today. So normally what you could do is just go over to exploration and use the faceted search feature to explore what different phenotypes are available either in their medical history or their diagnosis. But a lot of this is controlled access. So what I've done here is just kind of already gone to find data that I have access to when a user comes into BioData Catalyst. We've imported a few open access data sets, the 1,000 genomes, but also this tutorial synthetic data set. And we can, all users will have access to this. So this is a place where they can kind of start with the ecosystem and learn how to use it while they're maybe getting access to something on dbGaP. So then the next step that we'll do once we've selected our project is export all to Terra. This can take, just maybe 30 seconds or so. But what this is doing is, and again, if Gen 3 people are on the line or they might join in the question and answers with us later, is they're kind of packaging information about this data set into a PFB and sending that to Terra, and Terra can recognize it. And so what is being moved over is the phenotypic data as kind of text information that Terra can read, and DIRS links to the genomic data. So you're not actually exporting over large genomic files, but instead DIRS URLs that are pointing to buckets that contain that data. And what will happen is a new kind of Terra window will pop up. So this is part of the interoperability handoff. So we're going to be navigating between websites. So it's kind of hard to understand where in the ecosystem you are sometimes, but it's a really, I think, nice feature. So once you're here, you can have a few different options. You can start with a blank workspace, or maybe you've already created a workspace in Terra, you're a savvy Terra user. Or for the BioData Catalyst ecosystem, we have this other feature called Start with a Template. And this is similar to the tutorial I'm showing you today, where you can import your data directly into a GWAS. So again, because GWAS takes a really long time, I'm not going to import into the new one. I've already kind of done everything, so it's a little bit more of a baking show. So what will happen is it'll bring that, but clicking import into a template will bring you back to a space like this. And we can actually look at the data. So if you remember, there was many different nodes in the graph, and they're all represented here as different data tables in Terra. We kind of discussed the administrative nodes at the top, like program and project and study. You can see they're all represented by one. This might tell you, the researcher, like the name or the accession number. And then we moved down to go back to the subject, and this represents all the individuals in the study and their information. So we can click on the subject tab and open it up. And I just want to kind of highlight again what this looks like. So I think what an end user might be familiar with is if they open up the subject tab to find a subject ID from dbGaP that they're interested in, but instead they're finding this universally unique identifier. And so this is kind of a first like aha moment of what's kind of happening. But if we actually scroll over a little bit, you can see that the subject tab actually does have the subject ID that we're used to. It's just in a little bit of a different format. Another thing I just want to kind of point out is it's the same for demographic. So say we wanted to find that subject's actual information, like what piece of information here would we search in the subject tab? You could try to search for the submitter ID, but it actually doesn't come up in the demographic node. You could try to search this subject ID, and if you remember, the subject ID is one level up in the hierarchy. You went over to demographic and search that. You can actually find that that individual, that row, and understand their demographic data. Again, sorry, somebody's kind of being loud over there. If you don't mind muting your microphone. So again, just a little demo of what the researcher actually sees when they export the data. If you wanted to see exactly what when I was talking about how we're exporting over genomic data, we have kind of these pieces of metadata around the genomic data, and then we have this DIRS URL that can show us exactly where this data is located in what bucket. We're not going to use that today, so I just wanted to highlight it in case you had questions around it. So this is confusing, and so what we're trying to do for the end user in this tutorial is make code and Jupyter notebooks and things that can make it easier on the end user to interact with this new type of data model. And so one thing that we've done, and again, the dashboard kind of walks the end user through these steps, is we've created this set of Teradata utilities in a Jupyter notebook. Eventually, we might end up making this something like a pip install in a Jupyter notebook, but we wanted to keep it kind of open and transparent to the end user because we weren't sure of all the eventual use cases that researchers might want. And if they're savvy enough, maybe they could kind of play around with what we've created here to be able to kind of to make the data model that they're interested in using for their study. So again, this is going to just use, has a few merge functions. Either you can merge all the phenotypic data together, you can merge all the genomic data together, or you can merge genotypic and phenotypic data together. So what the researcher can do is basically run this notebook in a different notebook and call these functions that we've created for them to interact with this data. So that's this first consolidate Gen 3 data that you brought over. So we've kind of kept it in separate notebooks because, you know, Terra has a rich, you know, library of different types of analyses. And so maybe they could just export these few notebooks over to a different analysis if, say, they didn't necessarily want to do a GWAS and use this notebook and be able to kind of reformat their Gen 3 data for other use cases. So again, just quickly showing you that this notebook has a few quick functions to show the end user how to work with Jupyter Notebooks, how to work with Jupyter Notebooks, and in Terra specifically, like how the notebook interacts with these data tables that we're showing you, and functions to run this Teradata utils Python notebook, which can help us merge all of these different data tables that I showed you before together into something that's called a consolidated metadata table. So I'll just quickly show you the output of that. So we can come back over here to tables, and we have a new table called consolidated metadata. And this is a data model that the researcher might be a little bit more familiar with. The first column of this data model is the subject ID that they're used to understanding from dbGaP. And then, so you can move some screens out of the way. We have basically put all of the different clinical data together in this one kind of data model. So we've appended on the what data table these columns came from. So submitter ID is a repeated identifier in the Gen 3 model. So you can see that this column came from the sample data table. So they can import this, which is something they're a little more familiar with, into an actual notebook for preparing their data for an association test. So I know Geraldine kind of demoed Jupyter Notebooks yesterday, and I'll just kind of re-highlight some of the things that she showed. They're very nice. They're interactive, which I think is very important for the early stages of an association test. We kind of went over this before. This particular notebook was developed by Elisa Manning and Tim Majarian, and I've basically made it so that the way that we're importing this 1,000 genomes data in works seamlessly with the material that they've already created. It helps you understand how to set up your notebook. Yesterday, we kind of talked about how you can configure your notebook with a specific Docker. In this notebook, we're going to use this package called Hail, which is a Python-based package and is really very nice for working with large genomic data structures. VCF files, particularly from TopMed, are gigantic, and Hail is a way that you can kind of convert a VCF file to a different format and chunk it up into different pieces and then use it with a Spark cluster. So you can easily in these Jupyter notebooks customize and basically create a Spark cluster and parallelize across however many workers. And sorry, the Zoom thing keeps getting in my way. Yeah, and build the compute that you need. And the Hail team has some really nice materials around how to decide how much compute you need for the size of the VCF file that you're working with. So I'm not going to start the Spark cluster today because that takes time to load. But again, we have just some information to help the researcher really start up and get going. They're going to be learning not only the entire ecosystem, but the Terra user interface if they're not accustomed to it. So we're really just trying to lead the researcher through all of the steps that they need to understand to get going. So I'll just briefly scan through this, but you can see here that we're, again, using these functions we created to help bring in this data table into the notebook and turn it into a pandas data frame, something that might be a little more easier on the end user. And they can explore the phenotypes that come over from this project that they've exported. So in an association test, you need to, understand what the individuals look like and how, what the phenotype distributions are. So there's a lot of different examples here about how to visualize different phenotypes depending on kind of what you expect their distributions to look like. You can play with the data a lot, which I think is really nice and a great value in Jupyter notebooks. So again, I'm just skimming through this. We can subset that very large data frame that we created into actually what we want to test in our associations. So in this particular case, we'll be testing BMI and some covariates that we're including our sex, population, and LDL. So we're going to, initialize Hail. We have a lot of instructions about how Hail works and how Hail works with the Spark cluster. And we can bring in our cohort VCFs through we're kind of doing it in a different way here. Again, sorry, I'm not showing you exactly how the VCFs are coming over from Gen 3, but I could talk about that later in the discussion section if we like. And then, yeah, just like we explored the phenotype data, we want to explore the genomic data and account for anything that might affect our association test. So for example, if we have really closely related individuals, we need to make sure that we account for that with a genetic relatedness matrix, or if we have a lot of population structure, we need to account for that as well. So quickly moving through some of this code because I just want to kind of get down to the bottom. But again, you can see just how complicated an association test is. There's a lot of steps that the researcher needs to consider. And I think it's nice to have these markdowns to help the researcher kind of understand what to think when they're visualizing these outputs. So what we come out of this is we have a lot of different files that we want to import into our workflows. I can kind of move to the workflow section very soon and explain that. But basically we have, a file of phenotypes that has the subsetted phenotypes that we're interested in. We have potentially a new VCF where we've filtered for, say, only common variants. Maybe we don't have a high enough sample size to test for rare variants. We have the actual sample IDs because maybe we filtered out specific individuals because they had missing data and so on. So again, because the Terra user interface is, a little bit hard to get accustomed to, we're trying to show researchers how to create a new data table. And this is going to be called sample set, and it's going to include all of the files that we generated in this notebook. So, you know, I think a GWAS might be an iterative thing where you make a decision, you run the analysis, oh, it's confounded, I need to go back and reconsider. You can kind of keep a record of what you've done and what you've tracked, which I think is very nice. So here we're giving you code for kind of pushing these specific files and the saved Python notebook back to the data model. So it's called sample set. You can see I have already run it a few times because I needed to adjust things. I did something wrong and my workflows failed. So you can kind of keep record of different runs that you've done and the covariates that you were interested in using, what outcome you were testing, these links to the files in the bucket that you generated in this specific run. So we have the actual phenotypic information for all the individuals, and we've appended on some information about their genetic structure, their principal component 1 and principal component 2. And so we can easily kind of import this into our actual association test workflows. So these are workflows that we imported from DocStore. The nice thing about the featured workspace is that we can kind of already set up these workflows to work exactly with how we've kind of modeled how we're working with the data in Terra. So there's a lot of different ways that you could actually use the workflows, but I just wanted to show you how easy it is to link back out to DocStore. Again, this is showcasing the interoperability part of the ecosystem. And you could learn a little bit more about these workflows. You could go to the actual GitHub where the WDL is stored and learn more about it. The GitHub might have a little bit more documentation. There might be different versions and you want to check out a different version, or there might be some different files. They might have a JSON file in here that gives you an example. And basically, if you decide that you want to export a different version, or you've explored DocStore and found another workflow that you're interested in using, you can, if you're on a WDL workflow, you can easily export back to BioData Catalyst by selecting this button, and it'll export into Terra. If you're on a CWL workflow, it'll export to 7 Bridges. The same for the AnVIL. This button will export this workflow to the AnVIL ecosystem. Also, just quickly while I'm here, I wanted to highlight where we're actually keeping a lot of these workflows for these systems in this organizations feature. So we have a BioData Catalyst organization. We also have one for the AnVIL. And you can see this is where we're trying to share some of the workflows that we're creating for this ecosystem. And this is where we're also working with researchers to share their work. So another tutorial that is going to come in the future for us is a structural variant calling using graph genomes. And we have those workflows available here. So sorry for the diverge, but I'm going to go back to our analysis. So we're back in our Terra workspace. We're ready to start actual association test. This first workflow called BCF to GDS is going to change our BCF format to a genomic data structure format, because we're going to actually perform these association tests in the R language, and the genomic data structure is how the Genesis package in R interacts with the genomic data. So if we click on our workflow, our second one is the one I'm going to highlight. Again, I think this was brought up yesterday, how you can kind of set up your workflow in different ways. In this example, we're going to run workflows with inputs defined by data table. So if you remember, we made a sample set data table, and we can select the version of the sample set. So if we had different tweaks that we made to our notebook, we can select which one we want to use. And then how you do the actual attributes or inputs. Sorry, I'm drinking some water. Is that so if we're using the data table import, we will use this either workspace dot or this dot. So workspace dot means in the workspace section of our Terra workspace, we can pull in these specific files. It's a little confusing, but I'm going to focus more on this dot. So it means this data table, this sample set. In this sample set, we're going to take the phenotypes attribute. So again, I'll just open this up so you can see. So for this sample set that I've selected, the phenotypes column has the CSV file of phenotypes that I'm using. So it will pull this specific CSV file is how I'm directing the attributes here. And I can do this for a lot of different inputs. You can see there's a lot that we have to kind of import into this workflow. We're going to do a single variant association test. We can also, show the researcher a little bit how much disk and memory to ask for different parts of this workflow. And the other nice thing is that we can also direct the outputs for this specific workflow or this specific sample set that we chose back onto that sample set. So you have all of the inputs and all of the outputs in one place. So it's a nice kind of record keeper for what you specifically did in this analysis. So once I've kind of created new columns that will be added to the sample set data table by saying this and creating a new data table or column in that data table. So I've set all my inputs and outputs. I can just hit run analysis. It'll launch with Cromwell. Again, I've already done this because it takes time and I didn't want you to have to sit and wait. So I'm going to go down here and we've had a successful run. You can see the name of the sample set that I used, that it succeeded, how much it costs. We can explore it in this user interface where you can see the different steps of the workflow. We generated a null model. We performed our association tests, and then we summarized the outputs of this association test, and I'll show you that in a little bit, where you can create a quick figure to look and see if you think your association test was confounded or not. You can see the time it took for each step. So again, we can see the inputs and our outputs here. So we have all of the summary statistics, and then we've kind of summarized like the top summary statistics, or the top of the most likely candidates, and then you can just quickly view a ping. Again, this is a very downsampled tutorial data set, so these graphs are going to look a little bit funny. So this is a quantile plot that can help us understand if our workflow is confounded. And then this is a Manhattan plot, but it looks super weird because it only has a few variants on chromosome 10 and 11. So if you were using real data, this is where you might quickly want to check and see how your association went. Before I go back, I just also want to show you again how I told you that we were adding the results of our association test back to the sample set. So you can see that here, that there's new columns in the sample set ID with the files that we've generated from that run. So that's it. We performed an association study in the BioData Catalyst ecosystem. I just wanted to quickly talk about where we're going next. So we definitely want to provide more workflows in DocStore for all of our different projects, including BioData Catalyst and AnVIL, and trying to bring in Galaxy workflows in DocStore. The BioData Catalyst grant has a new fellows program, and these are early researchers that are going to try out our ecosystem, and they all have specific research use cases, and we're hoping to work with them so that they can share what they've created with the rest of the community and create tutorials a little bit like I did today that can really help new users get up and running in the ecosystem. I think the best way to make these tutorials is to pair up with research experts. It's kind of hard for us developers or even scientists that haven't worked specifically on this this type of analysis. We need a lot of input from the experts. Another thing that we really want to focus on is there's a researcher startup barrier to learning WDL and to learning how to use WDL, and we really want to try to pair a lot of the workflows that we have with a tutorial in Terra. And so that can either be like data preparation like you saw here today, or maybe you want to look at the outputs of your workflow. And then we're also focusing a lot on outreach efforts. I work closely with the DocStore team, and they've been putting on a lot of great webinars. There's a link here if you want to check out a DocStore webinar. And we're also hoping to have some workshops at Terra for when we're onboarding new BioData Catalyst users, and also kind of go to conferences. We have one planned at BOSC this year, but we'll see if that ends up happening with the current situation. And with that, I'm going to wrap up, and I think we have time for questions, or if you wanted me to focus a little bit more on a certain part of the analysis. Are you guys still there?
Yes.
Oops, sorry, I didn't hear that.
Any questions for Beth before we move on?
Not a question, more of a comment. I know it's a terrible thing to say, but that was a great presentation, a really great walkthrough. So thank you, Beth. That was very cool.
Thank you for sitting through it.
A quick question to go up several levels, back to the Gen 3 and Terra data, actually what you have on the screen now. As a researcher, or as you're working with researchers, where do they feel most comfortable doing those queries and doing that exploration and forming those cohorts? So forgetting about the downstream processing of the data, et cetera, but just the cohort formation. Where do you see that typically happening?
I believe that would probably be more in Gen 3. You know, you can, if you have a specific phenotype, I can also show you, I think, this is all open access data. I have the AnVIL version of Gen 3 available here. It's in staging. But Gen 3 has the faceted search feature where you can explore the data that is ingested in Gen 3. So in the BioData Catalyst version, they have, you know, around 80 top med projects, and it's difficult to understand what all data is available in those top med projects. So you can use their search feature to kind of understand that a little bit more and see what all projects you have access to. So I should hopefully in the future have more than just one project. So I could select 2 projects and export them both together into Terra, and then they would be, if they're in the same data model, they would all be in like all the demographic data would be here. So then if you consolidated everything, potentially you could understand the distributions of that a little bit more. But I think, is that kind of getting at where you're?
Yeah, exactly. The follow-on question is, again, not so much what's available in the system. There's a lot of things available in these different systems, but you know, from the researchers kind of boots on the ground, my follow-up question then is, do you see them launching notebooks to work with the cohort and fine-tune the cohort queries and selections away from the faceted search and more into programmatic access in a notebook environment?
Yeah.
Does that make sense?
Yes, we did that a little bit, and I don't think I spent a lot of time on it, and I don't think this is maybe the most elegant way to do this.
Yeah, you don't need to demo it or whatever.
Yeah.
I'm just kind of curious, you know, whether that
yeah.
happens in real life. That's all.
Yeah, definitely. I think, you know, you'd have no idea what the distribution of these phenotypes are, or, you know, necessarily like how to interact with them. So that, I think, is what this area of the notebook is for. It's like, let's explore actually what this cohort looks like, and I think there are issues and need to either add more data or think about it in a different way. I think that's what we've tried to provide here. I'm sure there are other resources that they could use outside of this, but this is
kind of my thought experiment is exactly this. You know, the kind of a rich exploration of the cohort formation and their characteristics, and then a rich exploration of the data that was processed out of that.
Yeah.
Thanks. Yeah. Nice to see it in real life.
As I have one specific question about using Terra. So, so when you select the input data, you can select the root entity type like something like sample versus sample set, that's different from, so then you can select the data and then there is a term set is used again. But those the set in the entity type versus select data, those are two different things, right?
Ooh, I'm not sure I understood your question.
Okay, I have a hard time to explain that too. So
You were talking about sample versus sample set?
yeah, I mean, you can.
Sorry, ladies. Sorry. Can I jump in real quick? Because I think
Yes, definitely. You're more an expert in Terra than I am.
I can clarify. So the select root entity stuff is kind of the older language that we don't use as much anymore. That when you have a workflow that is in the broad methods repository as opposed to doc store, there used to be a little menu. Well, there still is in that component a menu where you set the root entity type. Oh, I guess we still have that.
Yeah, sample set, and then if you click the select data part,
yeah, so
there will be the choosing existing sets. But
right.
this selecting data, this screen is happening both for sample and the sample set, but even they behave differently in the workflow, right? Depending on which root entity type I pick, but it's all still called set, right? Data set.
Yeah, it's really related to it's the terminology around sets that's a little confusing. The first one is really saying select the table that you're going to pull from.
Oh yeah, I wrote sample set to call this. Maybe that's the confusing part here. Set versus I'm calling it sample set. I think.
Possibly. No, actually, that makes.
No, that's the official term for participant versus sample, and then set for.
Yeah. So there's things. So when you create a set, it's because there's two different behaviors for working with sets. There's the thing where your workflow expects a set of things, which is basically an array, and you need to be able, like you select a single set and you give that to the workflow, and it's the one workflow submission that is going to take all the elements into of the array into whatever input variable is handling that. And that's the different behavior from if you have a workflow that expects a single file, for example, and you provide a set of entities so that each row is going to spawn a separate workflow, and the workflow is going to take in a single file from that list. And the thing is, you can use sample sets or whatever sets of whatever you want in both ways, and it really depends what the workflow expects. And I think right now there's a little bit of confusion around that because it's two very different behaviors. Like, are you giving the sets to the one workflow, or are you using the sets to spawn multiple workflows? Does that make sense? Does that actually?
Yeah, kind of. I mean, that's like, when I did a lot of trial and error, that looks like how it's behaving, but I wanted someone confirm that that's what it is, or I'm.
Yeah, it's because the root entity is the thing that's specifying what the workflow expects. Is really what it comes down to.
Okay, that makes sense. Yeah. Thank you.
Yeah, you're welcome.
Anything else? I mean, one thing I was hoping that this would kind of bring up a discussion around is how complicated this data model is. And, you know, for BioData Catalyst and the AnVIL, what, how we're feeling about that. And as we get into this NIH interoperability mindset and try to combine multiple data models, potentially what that would look like, especially to the end user.
Yeah, I would say that
exactly.
The data model is definitely something that deserves a lot of thought and coordination across groups, because that's probably the main obstacle to being able to combine multiple data sets. Because if you've got the one data set and some clear instructions of like, this is where this information is, you can figure it out. But once you try to combine multiple data sets, that's where it becomes a nightmare of different data sets.
Yep. Yeah, I can't like overemphasize that point. I don't want to say anything. I just like to hear other people talking about it. Thank you very much.
So we have a group. I don't know who's already working together, so apologies if this is redundant. We have a group that's working on kind of the next layer above, being able to say, here's a generic data model, like an abstracted data model layer, so that you could bring in different metadata structures and map that to a common data model layer to help with harmonization and searching across data sets. But yeah, anybody else want to share opinions and perspectives on that?
When I first was trying to use the Gen 3 data model, once I figured out that you're able to like step up, it made things a lot clearer. So I feel like it's not an insurmountable that we might be, or at least this is just my opinion, that we might be overestimating the difficulty of people learning the data model, but I completely agree that things will get very difficult when we try to combine.
Yeah. I can quickly just show you guys, because we have an example of, what's being made in the AnVIL, which looks like this, which is kind of similar to what I showed you, but not quite the same. So how would, if we wanted to compare, say, top med data from Gen 3, which has this data model, which is a slight variation off this, how will researchers combine these different parts of the data model together? How will they know how to map? And so I know there's discussions around that, but is the end user doing that, or are we doing that before the end user interacts with the data? Are we creating these models so it's easy to map across?
Isn't that
This is
what
Dave
Seven
Rogers.
Bridge is doing? That like each data is like has a unique ID, and then behind the scene they are doing harmonization. I don't know, maybe I misunderstood, but I think
So
what Seven is doing. No, yes. Not sure.
I didn't hear the end of that. Sorry, can you say it again?
Oh no, I mean just like that's a kind of like merging like different data model here. I might misunderstood, but yesterday I thought like Seven Bridge is doing that when they are incorporating different things that like each file is assigned with the unique ID, GUID, and then organize. Again, I could create the new combined data model behind the scene. I don't know. I maybe misunderstood, but
I'm personally, this is Garrett from the University of Chicago. I'm personally unclear as to the specifics of what SB might be doing. As we're looking at both NIH interoperability questions around having various data models and addressing the needs of specific projects like the AnVIL, which actively will have multiple data models for data just within that project itself. What we're partially leaning on is working on the front end to link data to external ontologies, such that even though the graph model for one program or in this case one system might be different from that in another, when we send data to an external system for analysis, or even within Gen 3 for analysis, we can rely on those linkages to external ontologies where available to run analyses on data that have two separate models. That is, one item might be 3 down in the graph for BioData Catalyst, and the same concept might be 2 steps down in a graph for AnVIL. And being able to send that data to Terra or to another platform and still run an analysis on it due to that linkage. I think it's probably the case for the other groups on this call, but it's certainly early days in addressing these issues because they are kind of novel as the number of commons and ecosystems and interoperable ecosystems have popped up. But that's the direction that we focused on so far.
I could probably provide a little bit of input on the 7 bridges side. So I think where maybe the similarity is that our data browser is also a graph-based query model. I think what we have done though is added a layer on top to kind of guide the user through the search, and it is very complicated. But I think we've hidden a little bit of the complication by having a sort of way for the user to sort of start at a certain point and step through things, which can also be confusing. Like if the user doesn't want to start at that point, it's more confusing. But it is a graph underneath, and we do have like a slightly different way of querying that graph, which is where I think it's a little bit different.
What I'd be curious about is hearing less about you know, the elegance or mechanisms to query the resulting graph. I think what we've mentioned earlier is the task of harmonizing and normalizing across 80+ projects, et cetera. You know, if anybody had any comments on that, or at least recognition, you know, what are our goals there over the coming year, et cetera.
So in BioData Catalyst, the data represented in this graph model, specifically for the phenotype data, has already been harmonized across all of the 81 TopMed projects, and it's been harmonized by, you know, a very large group of experts, the TopMed Data Coordinating Center, and then they've provided that, you know, to both dbGaP and Gen3. So you can easily compare maybe like around 50 harmonized variables, but you know, and that could provide interesting phenotypes to explore for association studies. It might be more that those variables are used as covariates rather than the actual trait that they're testing. However, there's a lot of unharmonized phenotype data for a lot more complicated disease phenotypes, and that is in a different location in the graph. And that's you know, even more of a problem. I don't necessarily think that's a problem that we as AnVIL developers or BioData Catalyst developers can solve, is understanding how to harmonize actual disease phenotypes in a scientifically accurate way. There's like working groups in these research or these researchers that work like in TopMed, for example. You know, they have whole working groups to try to solve these problems. So I'm not exactly sure what the plans are with the AnVIL. I could see us harmonizing you know, file formats and things of that nature, but not actually harmonizing the phenotypic data as well as like something like a data coordinating center could do, or we're only focusing on a few types of traits that might be easier to harmonize. But again, I'm not part of that harmonization group, but that's my understanding from the difference that BioData Catalyst has in terms of their harmonized data.
Perfect, thanks.
All right, well, I can give everyone a 4-minute break before the next talk if we want. I'm happy to sign off. Thanks everyone for listening and the great conversation.
Thank you.